For the past 8 years, I would say, we've been working with another group in Israel trying to reprogram cells of other tissues into beta cells. And this project is called reprogramming or transdifferentiation, because in addition to educating stem cells into becoming insulin-producing beta cells, we now have the ability of using a shortcut, using another cell type and turning it in one step into the cells that we want. The pancreas is made of acinar tissue, which is the one involved in the digestion of the food, and then the islets. And the islets are only 1 or 2% of the pancreas. 90-something% of the pancreas is acinar tissue or exocrine tissue. Every time we do an islet transplantation and we isolate islets, we throw away the exocrine tissue because it's no good for clinical therapies. We want only the islets. What if we could turn that tissue that we are throwing away into beta cells? As we speak, we are conducting the first transdifferentiation or reprogramming experiments directly into human acinar tissue, which is another tissue that not many other places have access to. But here at the DRI we do. Every time we do an islet isolation, we have plentiful supplies of this acinar tissue. So we are experimenting directly on that tissue. And even though this is very preliminary, the very first experiments that we did show that we have insulin production. So we are very excited about that. It's the very first experiment. There's a lot of room for improvement, but I'm sure that next year I'll be able to report even more exciting findings.